told you that it's an, obligate anaerobic bacterium. And I told you that it causes gas gangrene, which is the wound start to grow. And in its metabolism, during its metabolism, it produces gas, and gas just accumulates in the tissues that we have at the site of infection. And you see that if you see a patient who's suffering from gas gangrene, you see that the infectious site is inflamed, and this inflammation it's because of the the gas which has been built up in the infectious site. Okay, so in order to breaking down those large molecules, bacteria need degradative enzymes like protease, like lipase. Protease is able to break down proteins. Lipase is able to break down lipids. So degradative enzymes are also destroying the tissues. Also, these degradative enzymes, as they are destroying the tissues, help bacteria to spread into the tissues. And sometimes pathogenic bacteria, sometimes they produce toxins. You can see the definition of toxins here. Toxins are bacterial products which can directly harm tissues or trigger destructive biological activities. So some toxins, they can just kill the cell. Some just change the normal function of the cell. We have three types of toxins. We have endotoxin or LPS, we have exotoxins, and also we have superantigens. You already know some facts about endotoxin or LPS. What do you know about endotoxin? What do you know about endotoxin? When bacteria destroy. Which type of bacteria? Part of the cell wall. Gram positive or gram negative? Gram negative. Good. So we talked about endotoxin or lipopolysaccharide or LPS. Um, I told you that in gram-negative bacteria, we have outer membrane. And in outer membrane, we have endotoxin or lipopolysaccharide or LPS. I told you it is very toxic. If it is released in the body, there will be some severe outcomes. Even you can lose your patient if your patient suffers from a gram-negative infection. If you don't treat your patient, quickly, it's possible that the endotoxin, the concentration of endotoxin in the body increase. And as I said, there will be some severe consequences, outcomes. And death is one of the consequences, one of the outcomes of high concentration of endotoxin in the body. Okay, so here again you can see the structure of endotoxin. As I told you, there are three parts of endotoxin: lipid, A, core polysaccharide, and O antigen. So I told you that the toxicity of the endotoxin is because of the lipid A part. And when gram-negative bacteria are growing inside the body, it is possible that parts of the, their cell wall gets released into the body. So it means that endotoxin gets released into the body, and it actually stimulates the immune system to produce some cytokines, including interleukin-1, interleukin-6, TNF-alpha, and other cytokines. So interleukin-1 is related to causing fever. Okay, so interleukin-1 increases the temperature, the body temperature, and causes fever. Also, when endotoxin is released in the body, it causes leukopenia, which is followed by leukocytosis. Leukopenia means the low level of leukocytes, and it's temporary because after that we see leukocytosis, which is the increase in the level of leukocytes. And also endotoxin activates the complement system. Complement system is part of the immune system, and complement system are proteins which can destroy bacteria. So it makes sense. Okay, so the endotoxin is released in the body, so it means that there is this infection inside the body. So the immune system is responding to this infection. Part of its response is activation of the complement system. And also it can cause thrombocytopenia, which is the low level of platelets. And endotoxin, which can lead to a very serious condition, which is called disseminated intravascular coagulation, or DIC. Endotoxin, when the concentration of endotoxin in the body is high, it can lead to form small blood clots in blood vessels. And these, blood clots interfere with the perfusion of organs. And that's why the disseminated intravascular coagulation, or DIC, is very dangerous. Also, endotoxin, it can lead to hypotension, which is the low blood pressure. And when we have low blood pressure, it interferes with the perfusion of organs. And if the blood pressure is very, very low, it can lead to septic shock. Septic shock. So in septic shock, we have multiple organ dysfunction. And the reason is this impaired perfusion of organs. Essential organs like kidneys, heart, and brain are very important. If there is something wrong with the perfusion of these essential organs, there will be severe, outcomes. So in shock, or in septic shock, we have this multiple organ dysfunction, which is the result of this very low blood pressure. And if you don't treat your patient as soon as possible, it's possible that you also lose your patient. Antibiotics can cause endotoxin release. Yes, it's possible that you have a patient who is suffering from a gram-negative infection. You prescribe antibiotics, and the antibiotics are killing bacteria, and parts of the cell wall, which includes the endotoxin, are getting released into the body. So now we have suddenly we have this large amount of endotoxin in the human body, in the body of your patient, which can lead to, endotoxin. But you need to manage this situation, okay? You need to treat your patient. Supportive treatment are necessary in that case. If the, patient is showing the symptoms of septic shock, it means that the situation of your patient is really critical. So you need to address the situation very soon. Otherwise, you will be losing your patient. So another group of toxins are exotoxins. We talked about exotoxins in lecture number 1. I told you that some gram-positive and some gram-negative bacteria, they produce exotoxins. And we have two types of exotoxins. One group are cytolytic toxins. As you can judge by their names, they are able to lyse or kill the host cells. So exotoxins. One group of exotoxins are cytolytic toxins. For example, we have membrane disrupting enzymes in this category. As you can judge by their names, they target the cytoplasmic membrane of host cells. And you already know that the cytoplasmic membrane is super important for the cell. It doesn't matter if it's a eukaryotic cell or if it's a prokaryotic cell. It doesn't matter. Cytoplasmic membrane is very important for the cell. If there is something wrong with the cytoplasmic membrane, the cell will be dead. Even if the cell is a cell that we have in the human body. So membrane disrupting enzymes, they actually target the cytoplasmic membrane. Example is alpha toxin, which is produced by Clostridium perfringens. This is the third time that we are talking about Clostridium perfringens. I told you it's a gram-positive bacterium. It causes gas gangrene. And when it enters into the wound, during its pathogenesis, it produces, alpha toxin, which is the phospholipase C, and it targets the phospholipids in the host cell, destroys them, and finally, this toxin, the alpha toxin, is able to kill the host cell. Another group of cytolytic toxins are pore-forming toxins. They are able to form tiny holes or pores on the cell. And through these holes, ions and also water can leave the cell. So these toxins can interfere with the normal function of the cell, and sometimes they can lead to death of the cell. Example is streptolysin O, which is produced by Streptococcus pyogenes. Streptococcus pyogenes is a gram-positive bacterium. It can cause a strep throat. Have you ever heard the name strep throat? A strep throat. A strep throat happens because of the infection by Streptococcus pyogenes. Mostly children um, who are school age, um, they develop strep throat. It's a type of pharyngitis, and this bacterium can produce a streptolysin O. Another group of exotoxins are AB toxins. Yes, it's a contagious disease. It can spread through coughing. So another group of exotoxins are AB toxins. They have two main parts, A and B. A stands for action, and B stands for binding. Exotoxins are categorized as two types of exotoxins. Some exotoxins, they just alter the normal function of the cell. But some of exotoxins, they just kill the cell. Okay, so we have these two types of exotoxins. Through their B part, AB toxin just attaches to a receptor that we have on the host cell, and then the exotoxin enters into the host cell, and the A part gets released, and the A part is responsible for the action of the toxin. Sometimes this A part is responsible for killing the cell. Sometimes this A part is responsible for changing the normal function of the cell. Okay, so based on the type of the exotoxin, we have various outcomes, and I'm going to give you some examples of these AB toxins. So let's just start Vibrio cholerae. We talked about this bacterium. I told you it causes cholera, and Vibrio cholerae enters into the body through ingestion, and it produces cholera toxin in the intestine. So you're seeing the cholera toxin in this image. It attaches to its receptor. The A part gets released into the cytoplasm, and it actually increases the adenylate cyclase activity. Adenylate cyclase is a type of enzyme in the cell which is responsible for, converting ATP into cyclic AMP. So it means that now we have this high concentration, higher than normal, high concentration of cyclic AMP inside the cell, and it leads to diarrhea. How? Because when we have this high concentration of cyclic AMP inside the cell, electrolytes and also water, they just leave the cell and enters into the lumen of the intestine, and the result is diarrhea. And I told you that Vibrio cholerae causes watery diarrhea, severe watery diarrhea. So this type of toxin, cholera toxin, as you can see, it's not able to kill the cell. It's just changing the normal function of the cell. And we have another toxin, which is called diphtheria toxin, as you can see in this picture. It is produced by Corynebacterium diphtheriae. Are you familiar with Corynebacterium diphtheriae? It's the causative agent of diphtheria. Let me type it for you. It increases the activity of adenylate cyclase or its concentration. You see, this image is from Murray's book. It says increased adenylate cyclase activity. So all the images that you can see in my slides, they are from your reference book, which is Murray's Microbiology. So anyway, let's get back to Corynebacterium diphtheriae. It's the causative agent of diphtheria. We have two types of diphtheria. We have respiratory diphtheria, and also we have cutaneous diphtheria or wound diphtheria. The dangerous one is the respiratory diphtheria. It's a type of pharyngitis. The patient who's suffering from respiratory diphtheria, they have the infection in their pharynx, they have pharyngitis, they have fever, and sometimes because of the inflammation in the pharynx area, it's very hard for them to breathe. And it's possible that you lose your patient because this inflammation actually interferes with breathing or swallowing. And the toxin which is produced by this bacterium can cause effects on nerve cells and also heart cells, which can lead to death. So respiratory diphtheria can be fatal. So it's a very important and serious disease. So let's get back to its toxin. It produces a diphtheria toxin. Diphtheria toxin is an AB toxin, as you can see. Because of its B part, diphtheria toxin attaches to its receptor. The A part gets released into the cytoplasm, and it actually affects the protein synthesis in the cell. It stops protein synthesis in the cell. And you already know that protein synthesis is a very important function of the cell. If there is something wrong with the protein synthesis in the cell, if the protein synthesis is stopped, which means that the cell will be dead. So diphtheria toxin is a toxin actually which can cause cell death. No, it's not meningitis. Diphtheria is something different from meningitis. In meningitis, we have inflammation of meninges. But diphtheria, as I told you about the respiratory diphtheria, uh, it's pharyngitis. A severe pharyngitis, the patient has fever, the patient, pharyngitis, yes, not meningitis, pharyngitis, yes. How about the others? Do you hear me well? Is there something wrong with the voice? Do you hear me well? Okay, so it seems that others don't have any issues. Okay, good, thank you. Okay, so here I'm going to tell you more about other types of AB toxins. Okay, so again, we have Clostridium tetani here. We have Clostridium tetani. It's the causative agent of tetanus. You already know that tetanus is severe muscle contractions, spasm. It causes muscle spasm. And this bacterium actually produces an AB toxin, which is called tetanus plasmin, which affects neurons. Okay, so as you can see in this picture, tetanus plasmin actually blocks the release of inhibitory neurotransmitters. And that's why we have this continuous stimulation of the muscle. And that's why we see severe muscle contractions, which is very painful. So this is basically how tetanus plasmin works. It affects the neurons. And it's not going to kill the neurons. As you can see, it's just changing the normal function of the neurons. It just blocks the release of inhibitory neurotransmitters. On the other hand, we have Clostridium botulinum. In Persian, we say Clostridium botulinum. So have you ever heard the name of this bacterium? It's the causative agent of botulism, or in Persian we say botulism. What do you know about this bacterium, Clostridium botulinum? Okay, so what do you know about this bacterium? Inhibits acetylcholine release. Okay. Can cause paralysis. It can cause paralysis. Good. It can cause paralysis, flaccid paralysis, I would say. Okay, so Clostridium botulinum, it can cause botulism. It produces an AB toxin. Again, this toxin affects neurons. As you can see in this image, this toxin, this AB toxin, actually inhibits the release of acetylcholine. And it means that a stimulation of the muscle is blocked. So no stimulation for the muscle from that. And because of that, the patient actually develops paralysis, flaccid paralysis. So again, the AB toxin produced by Clostridium botulinum is not able to kill the cell. As you can see, it's just changing the normal function of the cell. And the last group of toxins are superantigens. Superantigens are very small proteins. As you can see in this image, they are able to attach simultaneously to MHC class II molecules on antigen presenting cells and also T cell receptors on CD4 positive T cells. And superantigens stimulate these types of cells, especially CD4 positive T cell. So the stimulation of CD4 positive T cell is dominant, and a large number, up to some references say up to 20% of CD4 positive T cells get activated by the action of superantigens, and they start to produce and release a large number of different cytokines. So we expect cytokine storm. So we expect cytokine storm. Let me type it for you. Okay, so we expect cytokine storm. And this cytokine storm can lead to fever, rash, and shock. Fever is high temperature. What about rash? What do you know about rash? What do you know about rash? There are red spots on the skin. There are red spots. There are red spots on the skin. Okay. And about shock, shock happens when the blood pressure is very low and we have dysfunction of multiple organs. Example of superantigens is Streptococcus pyogenes, which produces two superantigens, erythrogenic toxins A and C. You already know Streptococcus pyogenes. I told you that it causes strep throat. In school-age children, some patients, after developing a strep throat, they develop scarlet fever because of the superantigens produced by this bacterium. This is how scarlet fever looks. The patient develops, red spots on their skin, and this is called a scarlet fever. The reason for scarlet fever is Streptococcus pyogenes, which produces two superantigens, erythrogenic toxins A and C. Zahra is asking me to explain the mechanism one more time. Okay, so the mechanism is the superantigens, they can attach to MHC class II molecule on antigen presenting cell, and also at the same time to T cell receptors that we have on CD4 positive T cells. Both of these cells, antigen presenting cells and CD4 positive T cells, they are part of the immune cells, And super antigens are special because they can attach to both of these their receptor, I mean the T cell receptor and MHC class 2 molecule on the antigen presenting cell. So because of this simultaneously attachment of super antigen, both of these cells get activated, and they start to release, cytokines. I told you that CD4 positive T cell is dominant. They start to produce a large number of cytokines, and we see some outcomes. The outcomes are fever, rash, and shock. Got it? It's not like that. Always bacteria need to enter into the host and then they produce their toxins. Sometimes, bacteria, they produce their toxins in the food that we eat. And in that case, we actually we can get sick again. So these toxins are called preformed toxins. For example, we have Clostridium botulinum. You already know this bacterium is a gram-positive bacterium. It causes botulism. Sometimes canned food. Let me type it for you. Sometimes canned food are contaminated with this, bacterium. They produce the toxin inside the canned food, and if we consume this contaminated canned food, we develop botulism. I mean, Clostridium botulinum is able to, produce the toxin also inside the body, but it's also able to produce its toxin in the food. And we have a Staphylococcus aureus. It's a gram-positive bacterium, and you will be learning about this bacterium in your future sessions. So Staphylococcus aureus is a gram-positive bacterium. Sometimes custard-filled pastries are contaminated with this bacterium. So Staphylococcus aureus produces its toxins inside the food, inside this custard-filled pastries. And if we consume this contaminated pastry, we develop food poisoning. So the thing is, Staphylococcus aureus is not able to produce toxin inside the body. When we consume the contaminated food, which is contaminated with the Staphylococcus aureus, this bacterium actually is destroyed by the action of the acid in the stomach. So the stomach acidity is able to kill Staphylococcus aureus, but it has nothing to do with its toxin, which is already produced in the food. So if we consume the contaminated food with Staphylococcus aureus, this bacterium will be killed inside the stomach. But the acidity has nothing to do with the preformed toxin in the food. So the toxin actually inside uh, the body cause food poisoning. And we have another bacterium, Bacillus cereus. It's a gram-positive bacterium. Sometimes rice is contaminated with the spores of Bacillus cereus. It's a gram-positive bacterium. If we cook rice, the spores of this bacterium are resistant. You already know the facts about spores. I told you that they are very resistant to harsh environmental conditions, including heat. So if we cook rice, contaminated rice, the spores of the Bacillus cereus won't die. And if we keep this cooked rice outside the refrigerator for a long time at the room temperature, the spores start to germinate and they produce their toxin inside the cooked rice. And if we consume this contaminated rice, we can develop food poisoning again. So as you can see, there are some bacteria which are able to produce their toxins inside the food. But for example, Vibrio cholerae is not able to produce its toxin in the food. Vibrio cholerae needs to enter into the intestine, and then when it's inside the intestine, it's able to produce its toxin. Okay, so when bacteria enter into the body, I told you that they need to escape the host defense. They need to hide themselves from the immune system, from the defense mechanisms. They need, sometimes they just fight back against the host defense mechanisms. Okay, so different bacteria have different strategies, and I'm going to give you some examples. We talked about the capsule. I told you that the capsule is the outermost layer of some gram-positive and some gram-negative bacteria. The capsule actually shield bacteria. It actually shield the antigens that bacteria have. And by shielding the antigens, it means that the capsule is protecting bacteria against the immune system. So the immune system basically can't see them anymore. And I told you that, most capsules are made up of polysaccharides, and polysaccharides aren't very good immunogens. So it means that they are not able to stimulate the immune system very well. So it means that they can just escape the host defense mechanisms. And it's very hard for phagocytic cells like macrophages to engulf or eat the bacteria which have capsules. So again, capsule, as I told you, is very important to protect bacteria against our immune system. For example, we have Neisseria meningitis, which is a very important gram-negative bacterium. It causes, meningitis. So Neisseria meningitidis is a gram-negative bacterium which causes meningitis, and it's a severe disease which can lead to death in a very short amount of time. So this bacterium has a polysaccharide capsule, and the capsule protects the bacterium from the immune system. Or we have a Streptococcus pneumoniae. It's a gram-positive bacterium. As you can judge by its name, it can cause pneumonia, which is lung infection. So it can cause pneumonia, which is lung infection. And again, this bacterium has capsule, which protects it from the immune system. Sometimes some bacteria have enzymes which are able to destroy antibodies. Antibodies are products of our immune system, and some bacteria, they have enzymes which are able to destroy the the antibodies. Example is Neisseria gonorrhoeae. I told you it's the causative agent of gonorrhea, which is a sexually transmitted disease. And this bacterium, is transferred through sexual activities. So in mucus, we have, antibody, but this bacterium is able to inactivate the antibody. And in that case, it can escape the antibody function. And sometimes some bacteria have intracellular growth. They grow inside the cell. By growing inside the cell, bacteria are hiding themselves from phagocytic cells, they are hiding themselves from antibodies, they are hiding themselves from complement system. It's not like that our immune system cannot respond to intracellular growing bacteria. That's not true. There is part of our immune system which can actually function, which can act against these, intracellular growing bacteria. But I'm just telling you that by growing inside the cell, bacteria are hiding from some parts of our immune system. For example, we have Chlamydia trachomatis here. I told you it can cause it causes sexually transmitted disease in human. So this bacterium has this intracellular growth. We have again we have a Streptococcus pyogenes. This bacterium produces the enzyme which is able to degrade or destroy complement system. So I told you that the complement system is part of the immune system which is able to destroy bacteria, but the Streptococcus pyogenes is able to degrade the complement system. And we have some bacteria which are able to kill phagocytic cells. Examples, you already know the examples. Clostridium perfringens, I told you it produces alpha toxin, and alpha toxin is responsible for killing the cell. And also we have a Streptococcus pyogenes, which is a gram-positive bacterium, and it produces a streptolysin, which again kills the cell. The last topic that I'm going to talk to you about in this lecture is human microbiota. You already know some facts about human microbiota. Microbiota is also known as normal flora. Microbiota are microorganisms which are living normally on the skin or inside the body of healthy individuals. So these are called microbiota or normal flora. The genetic composition of microbiota is called microbiome. When we study the microbiota of a particular body site, we realize that microorganisms in that particular body site are classified as core microbiota and secondary microbiota. Core microbiota are microorganisms which are shared by 95% or more individuals. And the rest of the microbiota is called secondary microbiota. You should know that the majority of the microbiota is composed of core microbiota, while a smaller portion of the microbiota consists of secondary microbiota. So the majority of microbiota is composed of core microbiota. And in core microbiota, we have, small, we have this, smaller diversity in comparison to secondary microbiota. So it means that in core microbiota, we have microorganisms which are responsible for some functions which cannot be done by other microorganisms. So that's why we have this. We don't see this large diversity in core microbiota. But in secondary microbiota, we have microorganisms, and these microorganisms are also responsible for some functions which are needed for the human body. But these functions can be done by other types of microorganisms. So that's why we have this large, large numbers of different species in the secondary microbiota. So when it comes to the number, we have larger number of microorganisms in core microbiota in comparison to secondary microbiota. But these microorganisms that we have in core microbiota, they aren't diverse. The microbiota in the human body, they live in, upper respiratory tract, they live in mouth, they live in vagina, on the skin, and also in gastrointestinal tract. The microorganisms that we have in our gastrointestinal tract, they are very important. They do different functions. Sometimes these microorganisms are responsible for breaking down the complex, carbohydrate that we have in our diet. For example, cellulose that we have in our diet, it cannot be digested by the human body. We don't have the enzyme which is responsible for breaking down the cellulose. So it means that we cannot just use the energy from the cellulose. But the microorganisms that we have in the intestine, they have the enzyme to break down the cellulose and convert it into small molecules. And now these small molecules can be used by the human body as the energy source. So that's why the gastrointestinal tract, the microorganisms that we have in the gastrointestinal tract, they help us with the digestion of food. And some of these microorganisms also they produce vitamins like vitamin K, and our body can absorb vitamin K and use it. So in this slide, you can see what microbiota do for the human body, and also you can see what the host does in return of the favor of the microbiota. So as I told you, microbiota, they do some metabolic functions. You know, for example, in the intestine, there are microorganisms which can help with the digestion of food. And the microbiota also stimulate the immune system. It helps the immune system to stay alert. So if pathogens enter into the body, the immune system can respond very fast and quickly. So that's why microbiota are important for the health of human. And also microbiota prevent the colonization with pathogens. Microbiota, they don't like other microorganisms to enter into the body. So they compete with those microorganisms and they just remove them if they can. And in that case, we can say that the microbiota is also protecting us against pathogens. On the other hand, bacteria provide microbiota with a place to colonize. The host actually provides nutrients for the microbiota. And also, as we have the immune system and defense mechanisms which protect us against pathogens, it means that the immune system is also protecting the microbiota indirectly. So indirectly we are also protecting our microbiota from other pathogens, because as I told you, microbiota don't like other microorganisms to enter into the body. So the question here is, microbiota are able to cause disease or not? The answer to this question is yes. If microbiota leave the site where they are supposed to be, and when they enter into the sterile body sites, yes, they can cause disease. So they are safe as long as they stay where they are supposed to be. But when they enter into the sterile body sites, they can cause the infection. And also, sometimes when there is this disruption in the balance of microbiota, the situation which is called dysbiosis, it can also lead to the disease. Example is antibiotic-associated colitis. Colitis is the inflammation of the large intestine or colon. This colitis is caused by this bacterium Clostridium or Clostridioides difficile. And, sometimes this bacterium colonizes the hospital environments. And the spores of this bacterium are very hard to eradicate. Okay, so it's very hard to eradicate the spores of this bacterium if the hospital is contaminated with the spores of this bacterium. So it's possible that the patients who are hospitalized in that hospital, they get the Clostridium difficile from the hospital. If the microbiota in the intestine is healthy and normal, and if there is no dysbiosis in the microbiota of the intestine, when the spores of this bacterium enter into the intestine, the microbiota of the intestine, they just fight back against this bacterium, and they just remove this bacterium from the intestine. And it means that Clostridium difficile is not able to cause infection or colitis in the intestine. Okay? But, sometimes it's possible that there is something wrong with the, balance of the microbiota in the intestine. For example, sometimes it's possible that patients are on antibiotics. Antibiotics are, prescribed for patients to cure their infection. Okay, but one of the side effects of antibiotics is that they also have negative effects on the bacteria that we have in our normal flora. And it is one of the most important side effects of antibiotics. Okay, so I should say that the antibiotics, they upset the microbiota, and some patients, especially if they are on antibiotics which have this broad spectrum of activity, especially for a long time, to cure their infection, it's possible that they also lose a very important part of their microbiota in the intestine. And if this happens, it means that now there aren't very large number of microorganisms in the intestine to fight back against this pathogen, which is Clostridioides difficile. So if the spores of this bacterium enters into the intestine, now there is this space for this pathogen to grow and attach to the tissues in the intestine and cause the infection, cause the colitis. And this type of colitis, because it's related to antibiotic, is called antibiotic associated colitis. So it happens following the using antibiotics. So the patient who's suffering from this type of colitis, they have diarrhea. As a physician, you need to prescribe other antibiotics to treat this type of colitis. And sometimes the infection does not respond properly to the antibiotics. And in that case, it's possible that your patient, has the colitis for a long time. And it's possible that also you lose your patient. So sometimes it's very challenging for physicians to treat this type of colitis. It's possible to lose their patients too. But there is this, treatment strategy, which is called a stool transplant. The whole idea of, using a stool transplant is that if we are saying that dysbiosis is the reason for antibiotic-associated colitis, it means that if we return the balance to the microbiota, it's possible that they fight back against this pathogen and remove it from the body. So for this purpose, you get the stool from a healthy person. It's better to be one of the close relatives of your patient. You just prepare the stool. It's a very simple procedure, and you inject the prepared stool from anus to the intestine of your patient. So we expect that the microorganisms in the stool, they just start to grow inside the intestine of your patient, and they, it helps the microbiota of your own patient to get its balance back. And when the microbiota is normal again, they can start fighting against Clostridium difficile, and they just remove it from the intestine. And after a while, you see that your patient is improving, and you can save your patient by this simple procedure. Sometimes there are some special capsule. You can fill them with the stool, and you can ask your patient to ingest them. If your patient is not comfortable with that procedure, you can try injecting through anus. And if your patient, again, is not comfortable with either of these strategies, you can just grow the microorganisms that we already know that there are, which type of microorganisms we have in the soil. So we can grow them, we can cultivate them, we can grow them, we can propagate them in the laboratory, we can mix them together, and then you can, inject this mixture of microorganisms into the intestine of your patient through, anus. Okay, so as you can see, this is a very simple procedure, and you can just save your patient by using this procedure. So as I told you, we don't normally have this Clostridium difficile in the intestine. Some people, they do have Clostridium difficile in their intestine, but then the population of this pathogen in the intestine is under the control of the microbiota. And if something happens to the balance of the microbiota, for example, as I told you, because of the consumption of antibiotics, it's possible that they also develop colitis. Okay, so it's not always related to being hospitalized in contaminated hospitals. That's one case, but sometimes it's possible that patient already has this pathogen in their intestine, but it has been under the control. Now, because of the dysbiosis, they actually develop antibiotic-associated colitis. Okay, do you have any questions? Okay, so if you don't have any questions, um, okay. The course coordinator is Dr. Jabal Ameli. You should ask her about the number of sessions. I'm not sure about the number of sessions. You will be having 5 sessions with me. So there are already 2 sessions left. Yes, you can have the PDF files. Just you need to ask the CR to send me an email, introduce themselves, and I will share the slides with them. Okay. You have my email address. Again, this is my email address. You can take a screenshot. This is my email address. But please, only the CR send me the email, okay? I can just share the slide with all of you, just only with the CR. Yes, We have two sessions tomorrow starting from 8 a.m. Okay, so if you don't have any questions, enjoy the rest of your day and see you tomorrow. Goodbye.